Hello everyone, Mr. Linder here. Let's talk about the digestive system. So the GI tract is a long tube passing through the body, and its contents are part of the external environment. So when we put food and liquid into the digestive system, it actually remains in the external environment or the outside of the body until digestion occurs and we can bring that digested material into the internal compartments of the body. The primary function of the digestive system is to move nutrients, water, and electrolytes from the external environment into the body's internal environment. In order to do that, we have to be able to break down the large macromolecules, things like proteins, carbohydrates, lipids. We have to be able to break those things down and then bring them into the internal compartments of the body. So bring digested material into our blood or into our lymphatic system. So in order to break down things like carbohydrates or to break down proteins or to break down lipids, we have to have powerful enzymes. Okay? So there has to be enzymes in order for us to break down those large macromolecules, okay, things that are larger, right, into the smaller components that can be easily absorbed into our blood or into our lymph. Those digestive enzymes, though, are very powerful and can obviously digest parts of the human body. So digestive enzymes could digest the proteins or the lipids or carbohydrates that make up your own plasma membranes of cells. So at the same time that these enzymes are digesting macromolecules in the lumen of the GI tract, we also have to protect against autodigestion or digesting ourselves. So protective mechanisms have to be in place to prevent digestion of our own lining of the GI tract. If we were to digest the GI tract wall, we could possibly end up with ulcerations in the GI tract wall. So stomach ulcers, small intestinal ulcers, where these powerful enzymes have been digesting the mucosal lining of the GI tract. The GI tract also faces a mass balance issue as well. We have to be able to match the input of fluids into the lumen of the GI tract along with what the output is actually going to be. So it turns out that we ingest throughout the day roughly about 2 liters of food and drink. We also secrete large amounts of fluid into the digestive lumen throughout the day. So we have salivary secretions, we have bile secretions, we have gastric juice secretions, pancreatic secretions, and intestinal secretions. If you were to add this all up, there's about a 9-liter input into the digestive system. That's a large volume. So this large secreted volume has to be reabsorbed back into the body or we're going to have a dehydration issue. So if we don't take into consideration the amount of fluid that's moved into the gastrointestinal tract, we could have a dehydration issue. So how do we avoid dehydration in the human body when there's this large volume that's being dumped into the GI tract? Well, a lot of the volume is reabsorbed. Okay? So we have absorption along the small intestine and absorption along the large intestine, where we're taking a lot of that secreted fluids back into the body. So it turns out that on a regular basis, we're only looking at a small amount of excretion of fluids into our fecal matter, about 100 milliliters. And so there's only about 100 milliliter loss of volume throughout the day. So even though there's a large 9-liter input into the gastric lumen, we are taking a lot of that fluid back into the body to avoid dehydration. So reabsorption is actually very efficient in the human body, and we're only losing a small amount of fluids. Now, in some situations, vomiting, diarrhea, where you can have excessive water loss, that excessive water loss could lead to dehydration and ultimately failure of your circulatory system. Our circulatory system is dependent upon having the right volume within it. So if we're losing large volumes through vomiting or through diarrhea, then that can actually put stress on the cardiovascular system. Another thing that we should look at when we talk about the GI system is that we have to be able to repel microorganisms that can get into the gastrointestinal tract. Remember, along this tubing, as we move through the GI system, okay, and we're inside this tubing, this luminal space, the lumen is considered, again, the outside of the body. Okay, so that internal lumen of the GI tract is the outside. But in that luminal space, there are microorganisms. So the GI tract has a large contact area with the outside environment. And so as the body needs to absorb water and nutrients, it could also be absorbing microorganisms. And so the GI system has to be designed to repel bacteria, viruses, and other parasites so that they don't make their way into the inside of the body. So the defense mechanisms that we have in place in the GI system would be things like mucus secretion, enzymes, especially enzymes in the stomach or small intestine. We have acid production in the stomach. And then we also have a series of lymphoidal tissues along the GI tract called the gut-associated lymphoid tissue, or the GALT system. And some people call it the MALT system as well, the mucosa-associated lymphoid tissue. And this lymphatic tissue, again, is designed to protect us against microorganisms as they try to move from the external environment to the internal environment. When we think about GI processes, there are four main processes to consider when we talk about the digestive system. So the first major process is the idea of digestion itself. Okay? So digestion. So when we think about digestion, we're thinking about chemical and mechanical digestion. Mechanical digestion would be like you chewing up food, so mastication. Another example of mechanical digestion would be some of the muscle contraction that's taking place that's helping to mix up the food material. So mechanical contractions in your stomach or small intestine where you get mixing, segmentation, motility that's taking place. So those would be mechanical digestion processes. The chemical digestion that occurs is the enzymatic digestion. So using enzymes like salivary amylase or pepsin or trypsin or pancreatic lipase, using these enzymes to digest the macromolecules into smaller nutrients, smaller monomers, so that they're easily absorbed into the body. So that brings us to the next major process, which is absorption. So absorption is moving material from the luminal space. So this is the lumen, right? So moving nutrients from the luminal space across the epithelial cells and either into blood, or we could even add the lymph system. So as we bring nutrients across the epithelial cells, we're going to transfer those nutrients into the cardiovascular system or into the lymphatic system. And once they're in those systems, they can circulate throughout the body. So absorption can be an active process or a passive process to bring these now smaller monomers into the fluids of the body and be transported to other locations. The third main functionality is motility. With motility, we're talking about moving material through the GI tract. This is utilizing muscle contraction. Motility can be things like the migrating motor complex, peristalsis, segmentation. So we're looking at the smooth muscle contractions along the GI tract so that we can mix and move material that's in the luminal space. And then lastly, we have a process referred to as secretion. And secretion will involve moving water, ions from the extracellular fluid into the GI tract lumen. So we do have secretion directly into the lumen itself. That will allow us to maintain a certain water content within the lumen, so it's easier to move food material along the GI tract tubes. We also, though, have secretion of hormones into the cardiovascular system. There are a lot of hormones that regulate the GI system. So things like gastric inhibitory peptide, gastrin, secretin, lots of various hormones that must be secreted in order to have regulatory functions for the GI tract. And then we also have secretion of localized signals, what we call paracrine signaling that takes place so that we can locally control the GI system and have regulation over how the cells function in the tract. So there's a lot of secretion that is taking place in order to regulate what's happening along the gastrointestinal tract. Once we get nutrients digested in the GI tract and they've been absorbed into the body, then those nutrients can be used for cellular metabolism. So as we transport glucose, as we transport amino acids and fatty acids out to the body's cells, we can use those nutrients for the production of ATP. Or we can take those nutrients and store them for later use. So we could store triglycerides, we can store glycogen, and then utilize those stored nutrients at a later date. As you continue your studies of the digestive system, it would be a good idea to review the anatomy of the digestive system. So take some time to review the layering of the GI tract. You should be familiar with terms like mucosa, submucosa, muscularis externa, and the serosa. So this is the layering. So mucosa, submucosa, muscularis externa, and serosa. This is the typical layering that you will see in the digestive system. Also familiarize yourself with the major organs. Make sure you understand salivary glands and esophagus and stomach, the small intestine, large intestine, also those accessory type structures. So I mentioned salivary glands as an accessory organ, liver, gallbladder, things that are going to assist in the digestive process. So it's always a good idea to review your anatomical structures. As we move forward, I want to spend some time talking about motility in the digestive system. Motility involves moving food and liquid material from the mouth to the anus, and it also involves the mechanical mixing that works to break food into smaller particles. Motility is affected by the GI tract smooth muscle. It's also affected by chemical input, especially from the nervous system, hormones, and paracrine signaling. So there's a lot of, again, secretion of hormones and paracrine signals that are going to regulate the GI tract, and a lot of those hormones and paracrine signals are regulating the motility that takes place. The three main patterns of motility that I want to focus on in gastrointestinal motility would be the migrating motor complex, peristalsis, so migrating motor complex, peristalsis, and the idea of segmentation and segmental contractions. So let's start with the idea of the migrating motor complex. So in the migrating motor complex, what we're looking at is a pattern of motility that occurs between meals. So when you're, after you've processed a meal and you have essentially leftover contents in your stomach, small and large intestine, there needs to be this motility that takes place that will help to sweep the food remnants and leftover microorganisms through the intestinal lumens and make their way towards the rectum. So the motility will start in the upper portion of the stomach. So we're going to start the migrating motor complex in the upper portions of the stomach, and then those contractions will slowly sweep through the small intestine and then go through the large intestine as it sweeps material from one section to another. A series of contractions will take anywhere from 30 minutes to 90 minutes in order to get material swept from one section to another. So the migrating motor complex is really designed to take a long time getting material from one section of the GI tract to another and clean the digestive system as it moves forward. The next type of motility is peristalsis. Peristalsis occurs during and following meals to move food material in a directionality through the GI system. So essentially we're moving it forward through the GI system in wave-like contractions. So think of peristalsis as a wave of contractions and it's moving the material with directionality. So think of it as moving it forward. So as you swallow a food bolus, okay, so if this is your food bolus in the esophagus, there are peristaltic contractions that are going to move that bolus in a certain direction. The peristalsis occurs because of the circular smooth muscle. So there's circular muscle in the walls of the GI tract. And those circular muscles behind the bolus will contract. So we can see that in this diagram. There's contraction behind the bolus. And then the muscle that's in front of the bolus will relax. So the circular muscle that is ahead of the bolus is relaxing, and that will allow the bolus to move in a directionality, move forward throughout the system. This movement is faster than what you see with the migrating motor complex. You'll see anywhere from speeds of about 2 to 25 centimeters per second. Okay, so we're moving that bolus forward with a little bit of speed to get the material to where we need it to go. And again, this is regulated by hormones. It's regulated by the autonomic nervous system and paracrine signaling to make sure that we control the circular smooth muscle. So contract behind the bolus, relax in front of the bolus. And so again, we can see it again once the bolus has moved forward. Now we have contraction behind the bolus again, and then we're relaxing in front of the bolus so that we can move the bolus with directionality. So that's that idea of peristalsis. The third type of motility is what we refer to as segmental contractions. In segmental contractions, we are alternating the segments that are contracting. So essentially there's a churning that takes place within the intestines. So think of this as like mixing waves. And so mixing waves are going to help mix up the contents that are in the lumen of the digestive system. So if you look at the colors here, we have yellow, right? We have the yellow color, the green color, the purple color, and the light blue color. Okay? If we look at these various colors, as we have segmental contractions, we can begin to mix those colors together. So here we now see the yellow here, and we have the purple here, and we have green here. So there's this mixing that is taking place. And that mixing is going to allow better contact with the mucosal lining of the GI tract. You're going to get better enzymatic digestion of the macromolecules that are within the intestines so that we can better digest and then, of course, better absorb the monomers that are in the intestines across the epithelial cells and into the forms of circulation that we have. So either into the blood or into the lymph. So don't think of segmental contractions as having a specific directionality. Although there is some movement that takes place with segmental contractions, it's more about the mixing that takes place. So here we have circular muscle contracting, we have another circular muscle contracting, so we're mixing the contents together. Now, in some people, there are some motility disorders that occur. Motility disorders would range from things like esophageal spasms, constipation issues, diarrhea, irritable bowel syndrome, and a lot of these conditions are chronic conditions. And so people deal with these altered bowel habits on a regular basis, and they typically result in abdominal pain and nauseous feelings that take place typically after consuming some sort of food, but sometimes it's even at rest between digestive stages. So these motility disorders can be problematic at various times. Now, how do you control contractions in the GI tract? So where does this control come from? Well, the GI system can actually contract spontaneously. So there's control built into the gastrointestinal tract. Most of the GI tract is composed of smooth muscle cells that are connected by gap junctions and is controlled by pacemaker cells that are built into the gastrointestinal tract. There's literally, you can think of it as the brain of the gut. There's a lot of nervous system supply and a lot of signaling that takes place to control the GI system without the central nervous system impacting what is going on. Now, the nervous system does play a role. The autonomic nervous system will regulate things that happen in the GI tract. So there are sympathetic and parasympathetic control over the GI system, but a lot of the GI control happens at the cellular level, and it happens with pacemaker type cells. There are groups of contracting segments that exhibit different types of contractions in the GI system. And so I want to categorize those as tonic contractions versus phasic contractions. When you have tonic contractions, these are contractions that can be sustained for minutes to hours at a time, and typically occur in the smooth muscle of the GI system where you see sphincters. So where you have places like the pyloric sphincter, or whether you're looking at anal sphincters, these are contractions that can be tonic, where they can be sustained for long periods of time. There are also phasic type contractions, and these are contractions and relaxation cycles that last for only a few seconds. And so these are going to occur in places where we want to see peristalsis taking place, or we want to see segmental contractions taking place. So we either want to move things with directionality, or we want to see mixing take place. So how are these, though, contractions controlled? Well, cycles of contraction and relaxation are associated with spontaneous cycles of depolarization and repolarization. And so that's seen on this diagram, where we have depolarization and repolarization waves that take place here. And these waves are known as slow wave potentials. So slow waves in the GI system have a rate of anywhere from about 3 to 12 waves per minute. Okay, so there's about 3 to 12 of these cycles that happen per minute. If a slow wave does not reach threshold, then you would not get a contraction. So, for example, on this diagram, this is a slow wave that is sub-threshold. So we can see that right here. We have a sub-threshold depolarization that's taking place. If you have a sub-threshold slow wave, then you can see that there's no corresponding force in the musculature. So as these cells of the GI tract create these depolarization waves, they still have to be able to reach threshold in order to have an actual contraction cycle. So the more often that you reach threshold, the higher the rate of slow waves that you'll have. So sometimes you'll have situations where the rate is on the slower end because we're not reaching threshold as often, or you'll have rates that are faster because you are reaching threshold more frequently. You can see on the next one, this slow wave signal has depolarized to threshold, and so you're getting a slow wave cycle, and then we can see that there is a corresponding force of contraction along with it. We also see that if slow waves happen more frequently, we can have a summation of signals that occurs, and we actually can generate more force. So you can see that there's a greater force of contraction that occurs in the musculature. That greater force is going to be dependent upon the amount of calcium available within the smooth muscle cells. So when a slow wave reaches threshold, what essentially occurs is there's an opening of calcium voltage-gated channels. And when calcium voltage-gated channels open, calcium can enter into muscle cells, leading to further depolarization spikes. The repolarization is going to be controlled by potassium voltage-gated channels. When a cell fires one or more action potentials, then the calcium that is accumulating in the muscle cells can be used for muscle contraction. And then, of course, the contraction of the smooth muscle is graded. And so it's dependent upon the amount of calcium that's available and how much calcium enters the cell. A longer slow wave will result in more action potentials. Therefore, a greater amount of calcium is available to the smooth muscle cells. And if there's a greater amount of calcium, there's a greater force of contraction. So what are the cells that are generating these slow wave potentials? The cells that generate the slow wave potentials are known as interstitial cells of Cajal. So interstitial cells of Cajal produce these slow wave signals. And the interstitial cells of Cajal are basically a network of cells that are integrated with the smooth muscle cells. They are, in essence, modified smooth muscle cells. This is very similar to what you see in the heart. The heart, made of cardiac muscle cells, has integrated cells, part of the conduction system, known as the SA node and the AV node. And those modified cardiac muscle cells send electrical signals. So this is a very similar scenario to what you see in the heart. These are essentially modified smooth muscle cells that lie between the muscle layers and lie between the intrinsic nerve plexes, and these create the slow wave signals. The interstitial cells of Cajal essentially function as pacemakers, and so these cells will periodically generate slow waves to spread signals to adjacent smooth muscle cells, which are then interconnected by gap junctions. The fastest interstitial cells of Cajal are going to set the pace for the entire group. So if this is the fastest cell, right, of the group, then whatever rate it is sending slow waves at, that will be the signal for the rest of them. So if the rate is currently 5 slow waves per minute, then that means these cells are also, right, functioning at 5 slow waves per minute. So whatever the fastest cell is, that's going to dictate what the rate is for the rest of the cells. So these slow wave signals are progressing to the smooth muscle cells. So these are the actual smooth muscle cells for contraction. These are the modified cells up here, the interstitial cells of Cajal. These smooth muscle cells, upon receiving a depolarization and generating their own depolarization spike, will open more calcium channels. Those calcium channels produce action potentials, but that calcium is also leading to the contraction in the smooth muscle based on the interactions of actin and myosin within the smooth muscle cells. So that's a little bit on the digestive system and some of its basic processes and the motility that takes place in the digestive system. I hope that helps. Take care.